Evening viewer, welcome to our Glory Globe TV. Yes, like treasured recipes passed down from generation to generation, there are just some region of DNA that evolution does not bear POE. Mammals, fur, and birds share a variety of such encoded sequences, for example, which have remained untouched for millions of years. Humans are a strange exception to this club. For some reason, recipes long preserved by our ancient ancestors were suddenly spiced up with a short evolutionary period of time. Because we are the only species in which this region have been rewritten so rapidly. They are called human accelerated regions, that is called HARs. What's more, scientists think at least some HARs could be behind many of the qualities that set humans apart from their close relatives like chimpanzees and bonobos. Led by computational biologist Katia Pollard, director of the Gladstone Institute of Data Science and Biotechnology in the US, a team of researchers identified HARs nearly 2 decades ago while comparing human and chimpanzees genomes. In a new study, Pollard's team found the 3D folding of human DNA in the nucleus is a key factor in this pivotal moment for our species. Imagine a length of DNA from our last common ancestor with chimpanzees as a long scarf wrapped around your neck, with stripes of various colors running across its width down its entire length. Now picture someone tried to make the exact same scarf, but they did not quite follow the original pattern. Some of the stripes are narrower, some are wider, and some features color in a different order than the original. When you wrap the new scarf around your neck in the same way as the original, the stripes that sit next to each other in the loop are no longer the same. Like this scarf, a big difference between human and chimpanzee's DNA is structural. Large chunks of the DNA's building blocks have been inserted, deleted, or rearranged in the human genome. So human DNA folds differently in this nucleus compared with the DNA of other primates. Pollard's team investigated whether these structural changes in human DNA and its altered 3D folding could have led to particular genes with HARs being hijacked, linking them to different protein coding genes than they were originally applied to. Many genes with HARs are linked to other genes, acting as enhancers, meaning any increased transcription of their linked genes. Enhancer can impact the activity of any gene that ends up close by, which can vary depending on how DNA is folded, Pollard said. In a study published earlier this year, Pollard's team created a model suggesting the rapid variation appearing in HARs in all humans often opposite each other, turning the activity of an enhancer up and down in a kind of genetic fine-tuning, a model supported by their new research. For their most recent study, the team compared the genomes of 241 mammal species using machine learning to cope with a large amount of data. They identified 312 HARs are examined where they are located within the 3D neighborhoods of folded DNA. Almost 30% of HARs were in the region of DNA where structural variation had caused the genome to fold differently in humans compared to other primates. The team also discovered neighborhoods containing HARs were rich with the genes that differentiate humans from our closest relatives, chimpanzees. In an experiment that compared DNA with growing human and chimpanzee stem cell, 1/3 of identified HARs were transcribed specifically during the development of the human neocortex. Many HARs play a role in embryo development, especially in forming neural pathways associated with intelligence, reading, social skills, memory, attention, and focus. Traits we know are distinctly different in humans than other animals. In HARs, these enhancer genes, unchanged for millions of years, may have had to adapt to their different target genes and regulatory domains. Imagine you are an enhancer controlling blood hormone levels, and then the DNA folds in a new way, and suddenly you are sitting next to a neurotransmitter gene and need to regulate chemical levels in the brain instead of in the blood. Polarity. Something big happens like this massive change in genome folding, and our cells have to quickly fix it to avoid an evolutionary disadvantage. We don't yet understand exactly how these changes have impacted specific aspects of our brain development and how they became an integral part of our species DNA. Though Pollard and her team are already planning to delve into this question, but their research so far does show just how unique and unlikely the evolution of the human brain really is. The study was published in Science. BNS Report, Glory Globe TV, Delhi, India.